Hello everyone, welcome to today's live broadcast, Pre-emptive Genotyping for Pharmacogenomic Risk, Establishing a Benchmark. I am Christina Mahalik of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Affymetrix. Affymetrix, a part of Thermo Fisher Scientific, is a pioneer in microarray technology and a leader in genomic analysis. Affymetrix develops and provides innovative technologies that enable multiplex and parallel analysis of biological systems at the cell, protein, and gene level, facilitating the rapid translation of results into biology for a better world. We are proud to be part of Thermo Fisher Scientific, helping to advance our mission to enable customers to make the world healthier, cleaner, and safer. For more information, please visit affymetrix.com. We do have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We will try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit the problem through the green Q&A button found in the lower left corner. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Now I would like to introduce today's speaker, Dr. Ulrich Broeckel, Professor of Pediatrics, Medicine and Physiology, Chief Section of Genomic Pediatrics in the Department of Pediatrics, Medical College of Wisconsin. Dr. Ulrich Broeckel graduated from medical school at the University of Heidelberg, Germany. After residency training in internal medicine and cardiology, he carried out postdoctoral research at the Medical College of Wisconsin. He joined the faculty at MCW in 2000 and is currently professor in the Department of Pediatrics and the chief of the section of Genomic Pediatrics. His research focuses on the identification of genes for complex diseases, understanding gene functions, and application of genetics and genomics in clinical practice. His work combines clinical applications with functional genome analysis using induced pluripotent stem cells for disease modeling and risk prediction. Dr. Broeckel is the scientific director of a CLIA-CAP certified clinical laboratory, which conducts clinical testing for structural chromosomal abnormalities, as well as pharmacogenomic testing. I will now turn it over to Dr. Broeckel for his presentation.
Great. Well, thank you very much for the kind introduction. It's my great pleasure to have the opportunity to talk a little bit about the work we're doing in my lab, and we've been focusing on pharmacogenetics and clinical implementation of pharmacogenetics for many years. So I'm very happy and I'm very glad that I can give this presentation. And as it was mentioned before, if you have any questions, please type in these questions during the presentation. I will also give this presentation for our startup company, which focuses on preemptive pharmacogenetic testing, RPRD Diagnostics, which is a spin-out of the Medical College of Wisconsin and my laboratory. And so just a conflict of interest, we are a spin-out of the Medical College of Wisconsin, and part of the data generated here in this presentation was generated through an early access program, and that was provided by Affymetrix. So I want to tell you a little bit about pharmacogenetics and how we look at pharmacogenetics in the context of providing care to patients, adult patients and also pediatric patients. And I'll walk you through some of the examples, how we implemented and how we're contributing to the implementation of clinical pharmacogenetic testing in a preemptive setting. So I want to give you a little bit of background around pharmacogenetics in general. It's well established that the response to drugs in patients is actually variable. Not all patients who receive a drug respond in the same way. And a number of factors actually contribute to this. Obviously, there's the patient phenotype itself, the age, the gender, body mass index, diabetes, and so on, the characteristics of the disease. There's also, in many cases, it is actually not just one drug a patient received, there are multiple drugs. And so in many cases, we actually have drug-drug interaction, which actually affects the response to the variation of what we observe in a patient. And then obviously, the genetic risk factors and the genetic background plays a role, which affects then the response and the effectiveness of a drug. And this is really the area of pharmacogenetics. And all these things really combined, all these three major factors really determine the variation in the response in patients. And I want to talk a little bit about, in particular, pharmacogenetics and how we can use pharmacogenetics to tailor and provide the tailored treatment and provide better treatment to patients. So if you think about pharmacogenetics right now, in many cases, the clinical testing right now is very much drug-driven. We have a particular drug. It's very much candidate gene-driven. It's a single drug. We know, for example, a drug, and we know the target or the receptor or the enzyme which metabolizes the drug to make this effective. And so we're analyzing one drug combined with one gene. And in many ways, this is also then very reactive. So if a physician decides we decide to prescribe a drug, then we order a genetic test, and then in many cases that will drive then the way how we dose or the dose of the drug or what type of drug we would like to use. Now, if you think about this in general, so how pharmacogenetics has been around for quite some time, you can ask the question, so how many drugs are really out there where pharmacogenetics could actually or should be used in determining which drug is most effective? And because, I think there is well established now that there are a number of well-established examples that pharmacogenetic drugs and the relationship to the drugs actually maximizes the drug efficiency and also is able to reduce adverse events. In a recent review, if we just look, for example, at all the FDA-approved drugs, if you look at the lower bottom, about 7% of all the about 1,200 drugs which are currently approved, there is actually evidence for that pharmacogenetic genes actually affect the response. So you can look at obviously the number of drugs, but you could also look at the number of prescriptions which are actually filled in the United States, of roughly about 4 billion. About 18% of the filled prescription in the US have actionable, are associated with actionable pharmacogenes. And so if you look at all this combined, we have about 800 actionable genetic variants which are linked to the dosing and where we have guidelines from the FDA on these FDA-approved drugs. So it's not a small number of genes, and so that it is really a significant component of this. The other way how you can look at this is obviously you can say, if we look at particular genes which have been linked to pharmacogenetics, what's the frequency in different populations? And because of genetics and genetic variants have different frequencies in different ethnic groups. Here, this is an example of a recent review for pharmacogenetics. But here are the number of genes here on the bottom, a list of genes, and you can see that depending on the gene and the ethnic group, there for some of these drugs, there's a very high frequency of potentially actionable high-risk genes, high-risk genotypes for these particular genes. So all in all, the number of drugs and the frequency of variants for particular groups is actually reasonably high. We're going to the next slide. If we think about the disease spectrum, where pharmacogenetics can play a role, it basically covers all different diseases. So we see a lot of examples in psychiatry, in neurological diseases, oncology, has a significant number of genes and drugs which have been described, and particularly in oncology, the number of genes and relevant genes for drugs is actually continuously growing. But we also, it covers the area for infectious diseases, pain, cardiovascular disease, GI and diabetic disease. So it really covers the whole spectrum of all diseases where, main diseases where we're really focusing on. One of the questions we continuously get is, is this actually really, does it make sense from an economic point of view to do pharmacogenetic testing? Is this really cost effective? And I just want to pinpoint you to one particular article which was recently published in this year, which looked at the evidence whether pharmacogenetic testing in relation to particular genes and particular drugs, if this is really cost effective. And this review who reviewed very comprehensively the current literature basically concludes that they identified very robust evidence for the cost effectiveness of genotyping prior to the treatment for a number of quite commonly used drugs, not just very rare drugs, but really commonly used drugs. And obviously, additional research is really necessary to further expand on this, but it really already builds the foundation that it is actually, in fact, cost effective. So when we think about now clinically how pharmacogenetic testing is done. Now, pharmacogenetic testing has been around for a long time, and I gave you all this background here about pharmacogenetic testing. It might be actually, surprising then to say that, for many drugs where this is recommended or where we have really actually strong evidence that pharmacogenetic testing is beneficial, this testing is not done. And so in my presentation, we'll pinpoint to some of the reasons why this is the case. So, but currently, if we think about clinical pharmacogenetic testing, in many cases, this is really just done that a physician prescribes or wants to prescribe a particular drug. It's very much driven by a drug, very much candidate gene driven. We have a drug, we have a particular gene, and we're really combining just one single gene and one single drug. And in many cases, this is very reactive. So we want to prescribe a drug, we decide on the drug, and we immediately recognize then, we actually need this genetic test, and we order the genetic test, and in many cases then, you have to wait. And if you can't wait, then you sometimes prescribe the drug without pharmacogenetic testing. And so on this background here, we really thought about this. How can we, make pharmacogenetic testing, and how can we make pharmacogenetic testing more widely acceptable, and what is the type of infrastructure and approach what we actually could do to really improve the treatment and really bring pharmacogenetic more into clinical practice. And so if you think about all the genes now, all the drugs, what I told you, where we have this guidance or where we have really strong evidence, it's really a very limited set of established genes and established variants. And the other really important thing to keep in mind or to mention is that a lot of the genes which we use for pharmacogenetic testing are not linked to just one particular drug, but actually are linked to either a group of drugs or actually a large number of drugs, because these genes are, for example, enzymes in the liver which affect the metabolism, and so a lot of different drugs actually use a limited set of these enzymes. And so with that, if you have one particular genetic test, that actually becomes applicable to a large number of drugs here. And in addition to that, obviously, if you think about this, then with this limited number of set, if you do, then that's pretty inherent to any genetic testing. If you do pharmacogenetic testing, these results, because it obviously doesn't change over time, the pharmacogenetic testing results really retain their relevance. So if you test a particular patient and you get the results and you can determine that, then obviously these the results for this really retain a relevance for a lifetime. And so if you combine all this and you really think about that, it becomes pretty logical to think and conclude that really pharmacogenetic testing could be a prime example where preemptive testing actually really would make sense. So if a patient, an individual, has all the information available on these pharmacogenetic genes, you have the information readily accessible, you really can access this now at the time when you decide to prescribe a particular drug. So this is kind of like some of the background here and some of the concepts why we think that preemptive testing in the current time actually makes sense and is a very attractive approach. So there are a number of different initiatives which really started out potentially as a research project, but really bring this into the clinic and implement preemptive pharmacogenetic testing. My lab, we have been working for many years now with St. Jude's Children's Hospital in Memphis, and we collaborate on a project. It's called PGen for Kids, and it's really focusing on the clinical implementation for pharmacogenetics in patients at St. Jude's Children's Hospital. And so the experience, what we learned, is very much in collaboration with St. Jude's Children's Hospital. You can go to the link if you are interested more in this project. So the goal of this and of this really idea is that if we do preemptive pharmacogenetic testing, obviously we were interested and we need to develop a platform, a clinically testing platform, which allows us to analyze very comprehensively the number of genes. We need to develop consensus guidelines how these genes and the results are getting interpreted. Obviously, initially, as it started out with a research project, and we're going to continue to collect data. We recruit patients and families. And then the ultimate goal is to take these results from the clinical pharmacogenetic testing very comprehensively and integrate this into the medical record so that the information is accessible to physicians who want to prescribe a particular drug. And so I just want to give you one example for one particular gene, but in terms of the complexity, what goes into testing from the actual genotyping to the interpretation. And obviously, when we start with a DNA sample, you're just looking at one particular gene, CYP2D6. We obviously look at the genotypes itself. The genotypes then get translated in haplotypes as we group not only one single variant, but multiple gene variants to haplotypes. That then gets translated to a particular diplotype, in this case here. And based on the diplotype, we need to then translate this into the what we call the metabolizer status. So what is the activity of this particular enzyme? And then obviously, you can deduct from that the enzyme activity then determines how we want this particular, then how this affects a particular drug down the road. And so if you then, for example, a particular metabolizer, that for example could mean that if you can't metabolize a gene, or if you over-metabolize a gene, that you should either use one drug or you shouldn't use this drug. So it's not only, what I want to say here is not only that we have to analyze a particular gene to get the results, but there's a whole many more steps which really are necessary in the interpretation of pharmacogenetics until we get to the point that we have a interpretation and then a recommendation for a particular drug here. So let me start with the genotyping itself. Over the last few years, as we've been looked at a DNA analysis platform for pharmacogenetics for preemptive testing, which is very comprehensive. So we were one of a few groups who developed and established the Affymetrix DMET Plus platform solution as a clinical test, and it's a very comprehensive genotyping platform. Has about 1,900 markers on there and about 230 pharmacogenetically relevant genes, and translates these genes, what I showed you in the previous slides, to these star alleles, and allows you to analyze these samples and then get all the results for most of these variants. The platform itself was established quite a few years ago, and so very recently Affymetrix and Thermo Fisher now launched the second generation and update of this platform called PharmacoScan. And really the idea there is that it extends the DMET Plus platform and really is now much broader and really incorporates the results of the more recent publication and scientific evidence as it grew over the years. And now this platform has, you can see this here, substantial increase. We're now looking at about 4,600 markers in about 1,000, close to 1,200 genes. And again, translates to these star alleles. So just I want to review with you some of the content of the new PharmacoScan. We did early access and we established the characteristics of the platform, collected a lot of experience with this, and I'll walk you through some of the results now, what you can do, and show you some examples for particular genes. The genes itself are basically accumulate all the information from a number of different resources. One of them is CPIC. This is the Clinical Pharmacogenetic Implementation Consortium. The other one is PharmGKB, which is the Pharmacogenomic Knowledge Database. Those are both resources which curate pharmacogenetic information, and also then, in particular in the case for CPIC, information how you implement and guidelines on how to implement the results of pharmacogenetic testing. So here just a link to both of these sites. CPIC are an information consortium, if you want to follow this link, or PharmGKB are really two key resources for pharmacogenetics if you're interested in following this further and going deeper into this. So here's just roughly an overview of the different genes which are on the PharmacoScan array. So a number of CPIC genes here. These are all genes where we have guidelines for the clinical implementation, how the genotyping translates to a particular gene, how you implement then the interpretation with this. These are peer-reviewed publications, and you can obtain these articles on the CPIC website. There are a number of genes, mainly on the COIPs, where we call these haplotypes, these star alleles. I want to mention also that a number of genes, which is really very important for this platform, actually analyze copy number variation, structural abnormalities, and you can see this here on the right side. So it's a very comprehensive platform in the content of this. The assay itself, let me walk you through here real quick, is a 4-day assay. So we basically start with DNA, followed by an amplification, a fragmentation on the second day, and a precipitation and a resuspension, and on day 3, hybridization. These are the typical standard array-based methodologies for genotyping. And then on day 4 is a ligation and scanning of the chip itself. For the protocol on the right bottom, you can see the hands-on time. So overall, it's a pretty straightforward assay, performed really well. And on day 5, the key thing here is that, as I told you before, we not only have the genotype, but we actually provide a lot of interpretation with this. And so there is a analysis suite which actually allows you to perform this analysis, to translate this into star alleles. So let me show you just a few examples here, how the output the output you get from the software here. And you can see here, for example, here, this is just one variant where you see here the clustering for the homozygous allele and heterozygous allele. So you can see here how we get to the particular genotype. Here is a overall quality control. Quality control, particularly in the clinical setting, is very important. So call rate needs to be tightly monitored here, and you get summary reports on the test here. This is, again, here an example of the SNP clustering. You can see here the different the genotypes here. You can pick one particular gene, and so in the next slide here, we'll look at this. So I told you before, some of the genes which play a role here in pharmacogenetics, you have copy number variation. So you either have duplications or deletions of the gene, and you can think about that. If you have a deletion of a particular gene, then this is, for example, an enzyme. You have then reduced activity or no activity. So the analysis of copy number variation is critical for calling the correct genotype, not only on the SNP level but also on the copy number level. And so, as you can see here, it allows for the analysis of copy number variation, or, for example, or deletions here for this particular gene. That would be, for example, CYP2D6. There are a number of copy number probes included in this analysis. The other thing which is available on this platform here is the analysis of HLA class 1 and class 2. Here are some examples, a number of examples where HLA genotypes and haplotypes actually contribute to the risk. So that is an improvement compared to the previous DMET Plus platform as well. And the first question then, when we ever start a new platform, particularly when you want to translate this into clinical care, is really the performance and the characteristics. And so I'll show you here one slide where we, from our early access program, so we typed samples which are with established genotypes here, and we looked at the concordance rate, and we looked at the call rate, and the pass rate. And if you just look here at the bottom here, the most importantly is the concordance with the samples in the previously genotyped results for overall as well as for the heterozygotes. And you can see that the concordance from the previous platform to the common the PharmacoScan platform is over 99%. So we were quite excited to see this high concordance rate, both for samples from Coriolis, our internal samples, which we collected here. The other thing, and this is a resource for the pharmacogenetic testing community here, I want to draw a little bit attention to this. The Center of Disease Control actually has a program that's called the Genetic Testing Reference Material Coordination Program, or GeT-RM, very long name. And so the goal of this project is that we establish a set of DNA samples with known genotypes, pharmacogenetic genotypes, and so these DNA samples from cell lines are available from Coriolis. So if clinical labs are interested in establishing this, they can actually order the cell lines for proficiency testing and establishing the assay in their lab. And so we used PharmacoScan on these samples, and we actually contributed with DMET Plus to this program, number of different testing platforms here. Just a list of all the testing platforms which contributed to this program in the pharmacogenetic area, and contributed the genotypes to the website. And so we also used the PharmacoScan platform now to genotype this reference samples, and we'll provide the genotyping results on these samples for the PharmacoScan platform to this website for comparisons. So just looking at this very cursory, and we started to do careful comparisons, is that if we analyze the samples and if they have a high call rate, here over the call rate on these reference samples was over 99%, as you can see here. And if we look at the concordance then between PharmacoScan and DMET for this reference set, you can see here that the concordance was extremely high. I also want to pinpoint then that selecting quality control parameters is important, and we have, for example, if we have samples here where the quality cutoff, the call rate was not met, these samples then have lower concordances, and then obviously should not pass in a clinical testing environment. So that is as control here as well. So overall, so we have here the pharmacogenetic now testing platform to be very comprehensive. I think this is a prerequisite. And really then the idea here is, if we think about implementing pharmacogenetic testing, we'll start with then the physicians here who orders the test. We do in our CLIA approved pharmacogenetic lab. We perform the testing, we perform the genotyping, do the data translation, and then this is the next step here. It comes to the interpretation, not only to the particular haplotype or diplotype, but also then the interpretation on the metabolizer status for a particular gene. And then with these results, it goes back to the implementation to the physician then for the interpretation as it relates in for a particular drug or a drug combination. So really, this is kind of the circle where we see this. And so to the last point then, when we have these results, what's really important is that we take the genotyping results, the interpretation, metabolizer status, and really, I think because pharmacogenetics is so complex and there are so many drugs out there, and the interpretation really really needs and requires a lot of expertise. but it is really important that we really provide the additional decision support around this. And I think here this is where, in particular, the integration into an EMR becomes absolutely critical. And so I'll show you some examples, which were developed at St. Jude's in an EMR, Cerner as a platform to really help physicians manage pharmacogenetic information. And so here's just one particular example for TPMT, which is used as a genetic test for thiopurines. And so let's assume you want to prescribe, a physician wants to prescribe this, and we don't have any genetic results done in the EMR. You can have a decision support pre-test warning that pharmacogenetic testing is really recommended, and we don't have any results, and really direct the physician and the ordering physician to basically order this pharmacogenetic test before you prescribe a particular drug. Now, if you actually have preemptive pharmacogenetic testing, you can actually incorporate this already and in medical record and decision support here as well. And really, let's say, for example, you have certain results here, you can actually add this into a problem list, and so then the results are really available. For example, if you have a patient who is a poor metabolizer for a particular gene, and then you can develop this decision support to link these high-risk genotypes with drug ordering or prescribing information. And so here's one example. Let's say, for example, you have the information on, we go back to CYP2D6, and you do want to prescribe a drug which is linked to this particular gene and to the particular metabolizer status, the result of the pharmacogenetic testing, then really the decision support in an EMR can be very handy to basically, give physicians a warning, say like, maybe we really want to consider this because this pharmacogenetic testing is currently available. So this is this post-test infrastructure. And I think with this, if we really think about some of the hurdles what we have in pharmacogenetic testing is really the interpretation, the complexities around interpretation. Being able to automate that, I think, is going to be a key step to really allow us to make pharmacogenetic testing much more available in the clinic and make this much more straightforward. So we've been doing, not only with St. Jude's, a couple other groups, we've been doing preemptive pharmacogenetic testing. There are a number of other organizations out there as well who have programs and who do pharmacogenetic and preemptive testing. And overall, from our experience, I just want to summarize here, obviously, the pros and the advantages if you have this testing available. Basically, in our opinion, I think, in my personal opinion, one key advantage is that, really, if you have the genetic test readily available, you have these tests whenever a critical decision, important decision needs to be made. So do I prescribe drug A or do I prescribe drug B, or should I basically avoid drug C at all? So those are really, decisions which are made at the time, and so basically having available pharmacogenetic testing information just prevents the delay of like ordering a test, waiting for the result. And in many cases, you actually can't do this. You don't want to wait until you have a genetic test. And then, so I think that really is the main advantage. The other key thing here is, and this is really very much on the technical side, if you think about it, you have to analyze a lot of different genes and you bring this in the medical record. One of the concerns we always had is that, is this really expensive? We can't really afford that. Doesn't this make, this really you can't really overcome this. And I think one of the really, and not just in general, one of the really great, improvements we had over the last few years, particularly in the DNA analysis platform, is the tremendous amount of reduction and tremendous reduction in cost for genotyping and DNA analysis. And I think with this really very comprehensive pharmacogenetic testing comes very much into the realm of that you can do this, and a lot of patients can afford this, and the cost of these gene panels becomes very, very feasible, and compared to even just a few years ago of the cost, if you compare this to the cost of one particular genes. And then again, I think the improvement in learning and implementing EMR and the decision support in an EMR I think this really becomes much more widely acceptable, and so I think this is also a very critical component with this. And what we see in general is that if you have pharmacogenetic results available, and these are available to physicians, and you have the decision support, then it actually increases also the acceptance of pharmacogenetics. There is still a hurdle here, despite recommendations, despite recommendations by the FDA and guidelines, there is still a lot of hurdles here to implement pharmacogenetics. And I think preemptive results available, decision support, I think really we've seen is that this really increases the acceptance for physicians and reduces the overall hurdle to pharmacogenetic testing. Obviously, some of the challenges what we've experienced all along is that the genotyping itself, if you really think about this, the cost came down. The genotyping itself is really the easy part. It's really the implementation, which is still critical and still a challenge. And then it starts with basically everybody who's involved in the whole process. And so it's not only physicians, there's also pharmacists, there's nurses, there's patients, and so this really needs a very comprehensive approach. Needs the discussions with the healthcare organization, with physicians, to understand their needs. And so, if I could summarize some of the lessons we learned, just some of the key points here. What we certainly going to continue to learn as we implement different genes, as we implement new guidelines, as we expand now from the old platform to an even more comprehensive genotyping platform. In our experience, implementation is always site-specific. It really, as I mentioned before, it's very much driven by clinical needs, by the patient population, and by the overall interest of healthcare organizations. If you want to implement this, it's always important to capture opinion leaders in your organization to really drive this forward. Look at the needs, in your organizations. Education becomes key, collaboration with physicians, and really the continued evaluation of the scientific evidence is also a key component. And all this really, as I mentioned before, really requires a multidisciplinary team. So it needs the clinical lab, it needs the pharmacy, it needs pharmacogenetic experts, it needs the physician, and it also requires the input by medical records, EMR, and clinical informatics. So with that, let me summarize that we think that preemptive pharmacogenetic testing really offers a lot of clinically relevant and important advantages for the implementation of pharmacogenetics in the clinical care. As we move to now the PharmacoScan as a more comprehensive genotyping platform, we think that this really is a very cost-effective way to generate pharmacogenetic information, which builds the baseline for the integration into EMR and the more intelligent prescribing practice. The analysis software is obviously key. That really is important as this translates the haplotypes and the diplotype nomenclature. This is often complex, for not one gene it varies between all the different genes, and so this is really key. That is really well curated. And as I said, previously, we will actually generate and provide the generated genotyping results on these reference samples. We'll provide this to the CDC website for PharmacoScan as a resource to the scientific and clinical testing community. And finally, let me mention that our company actually offers this clinical pharmacogenetic testing and services and consulting around clinical pharmacogenetic implementation. And so with that, this has been an effort from many individuals for a number of years now, particularly my lab, research and clinical lab here at the Medical College of Wisconsin. We have very strong support, and we appreciate the support by Affymetrix and the people at RPRD Diagnostics, as well as my collaborators at St. Jude's Children's Hospital and at the CDC. And with that, I hope I could give you an overview of our approach and some of the concepts and ideas around clinical testing for preemptive pharmacogenetic testing. And I will be very happy to answer any of your questions. So thank you very much for your interest and your attention.
Well, thank you, Dr. Broeckel, for that informative presentation. It is greatly appreciated. And before we get started on the question and answer session, the interactive part of our presentation today, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found once again by clicking on the green Q&A button at the lower left of the presentation window. Remember, we'll try to answer as many questions as we can today as time allows. All right, so why don't we go ahead and jump on in with our first question. And the first question asks, do you have specific health economic models that demonstrate the value of reduced ADR due to preemptive genotyping?
That is a very good question. We get this. This is a very good question. We get this question all the time, and I think there are really good examples out there which actually show. And this is this review article I mentioned one of them. A lot of other studies are looking at this and starting to look at this, which actually showed that pharmacogenetic testing actually does reduce costs and does economically make sense. And then really the question becomes is that if you do this preemptively, is the cost for preemptive testing, in which comes and is associated with generating the results and translating and bringing this into the medical record, is this really cost effective? And if, as I said and mentioned before, the cost for genotyping, if you look at just a few years ago, what was the cost for one or two tests? If you just test one or two genes, that was a significant cost. And we're now at the level with the reduction in cost. What we see is that the cost what you have if you test 1, 2, or even 3 genes, that's about the same cost what you get if you do now with these new technologies. If you do the preemptive or if you do the genotyping, pharmacogenetic testing on a panel. So I think with that, that really makes a significant impact and makes a significant dent in this field, and I think will really build the platform and the foundation for bringing preemptive pharmacogenetic testing to a more broader application.
Excellent, thank you for that. Our second question of the day asks, do MDs fully understand how to use pharmacogenomic data, and what tools are out there to help them?
That is also a very good question, and I think it's fair to say that pharmacogenetics and the interpretation of the results, genetic results, how it translates to the activity of a particular genes, how this then links to a particular drug a physician would like to use, is really complex. And there are lots of drugs out there and there are lots of genes out there. And so I think for a number of pretty common drugs where the pharmacogenetic testing is well established, has been established for a long time, I think physicians really know how to interpret this. But the data and the amount of data is really large, and you can't really just memorize this. And so I think what's really important here is that we have infrastructure and decision support. And so there are a number of initiatives out there, a number of resources out there which really help and guide towards that. And I think this is really growing. And as the field is growing with pharmacogenetic testing, these tools are developed. I think a key component, as I mentioned, is really decision support inside an EMR. This is where a physician or a nurse, pharmacist, this is where the decision is made, what type of drug is used. And I think integrating the decision support into the EMR, providing this guidance, accepted guidance, peer-reviewed, support based on peer-reviewed support. That's really important. And there are a number of groups out there which really develop and drive this forward. And so I think that is really what I see is an important component in the implementation of pharmacogenetic testing.
And thank you once again for that answer. Looks like we've got some more questions. Our third question today asks, are there many different technologies for PGX testing? Can you talk about the best uses or pros and cons of each, please?
Yeah, so as you say, there are a number of different platforms out there, and different vendors are continuously developing new platforms. So we evaluate a number of different platforms, and so they're not criteria as how you can decide what you want to do. As I mentioned in my presentation, so if you look at PharmacoScan, that is a test which generates a lot of data. We're looking at very comprehensively a large number of genes. It takes about 4 to 5 days to really process this. So that you need to take into account if you order a test. There are other technologies out there which analyze less genes. Obviously comes with the advantage that you don't have to handle a large data set. And some of these platforms actually can get the results not just in a few days, but you can get the results maybe in as short as like a day, or even shorter. You can measure this like in hours. And so that really depends also on the clinical needs. Some genetic tests you need to have a very fast turnaround time. Most of these tests actually then analyze just a smaller number of variants. And then in other settings, you have a little bit more time. You can do then very comprehensive testing where you have maybe a more, a longer turnaround time. So I think that really kind of like plays into this. Cost is also an issue, and intuitively one would think that if you test one gene, that might be less expensive than if you test 10 or 15 genes. But as I mentioned before, the changes in the technologies, what we have is now that actually testing a large number of genes can be very competitive in terms of pricing compared to for example, analyzing just a smaller number of genes. So it also plays a role is like how comfortable healthcare organizations and physicians are with managing larger data sets, and so that could speak, for example, for a to choose a platform which has a limited number of variants. But turnaround time, I think, is a key component which drives this, and then the cost, and then. Finally, and this should be basically standard, is obviously like with any test, if you implement this clinically, quality, consistency, reproducibility, I think are key components and need to be evaluated in any platform you would like to choose.
Okay, our next question asks, why is preemptive screening more attractive than focused clinical methods?
Yeah, so this is a good question. As I mentioned before, there are a number of groups a few years ago where really kind of like they started preemptive pharmacogenetic testing as a research project, and really thinking about this. We think about genetics in general, and we're moving towards more and more genetic information. How do we deal with this pre-existing genetic information and the integration, and how does that really translate into clinical decision support? And so from our point of view, if you have time, and in patients, for example, who have a high likelihood that over either their lifetime or the next few years that they have a high need to get like different drugs, having the genetic information available and being able right at the time when a physician will decide to prescribe a particular drug, if this information is available, having this and then basically translating this immediately into the decision support, that should, by all means, should have, should make pharmacogenetic testing, pharmacogenetic implementation much more easily available and implementable. So we do really see that if the data is there and if the right decision support is there, that this really should make pharmacogenetic implementation more acceptable. And really, as I mentioned before, the cost plays a role, and the technologies really advanced so much that now more comprehensive testing is really in the realm of like being very price competitive compared to like the single genes. And I certainly see that there is much more interest now in preemptive pharmacogenetic testing. It's also really the idea of being able to have this data and physicians being comfortable with physician support, which really drives this. And last but not least, I think patients are also getting more educated about genetics and the information there, which is available, and the opportunities what genetic offers. And so I think we certainly see an interest from patients as well.
Excellent. Thank you for that answer. Looks like we're going to have time for at least 2 more questions. So let's begin. Have you seen trends with regards to labs moving increasingly towards preemptive genotyping solutions?
Yeah, that is a very good question, and I can definitely say yes. When we started, it was more of a challenge to really convince people that the concept of preemptive testing. It is still not generally accepted. That I think would be too strong of a statement, but we have definitely significant interest from a number of healthcare organizations, hospitals now, as the results and the experiences of a number of the groups who have been doing preemptive testing, this becomes available. And so I think there's really growing interest there, and I think with the growing interest, there's really then also the drive on the clinical testing side to really expand on this. And again, the new platforms, the reduction in cost, I think that's also a really important component into this. So if you need testing for, let's say, 2 genes, you use a more common, very targeted platform, and that costs as much as the clinical test as doing this a whole panel, very comprehensive testing. You get substantially more data. I think that also drives the interest in this area.
And this will be our last question of the day before we wrap up. Have you heard feedback to support that this approach leads to ADR reduction?
That's a very good question as well, and it really plays into the overall concept of, like, is this cost effective? What is really the outcome? And yes, we do obviously have evidence that the preemptive data is actually the pharmacogenetic results, which are in the medical record, are utilized actually quite significantly. That obviously depends a little bit on the patient population. But clearly this is a very high utilization here, and we do have evidence that having pharmacogenetic testing result available really very directly either looks at adverse events or really targeted and brings or improves the way how for example, physicians pick a particular drug, and so really reduces the time a patient either gets to the right dose or really finds the right drug for the particular disease. So, but this is still intense area of research, and we clearly see that as this approach even becomes more widely available, that then the data, which looks not only at cost effectiveness but also at the outcome and how it benefits patient treatment, that the body of data and results really will grow here.
Excellent. Thank you once again for not only your time today, but your presentation, Dr. Broeckel. I would also like to thank our sponsor, Affymetrix, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through February 2017. And you will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you and encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you so much for tuning in. This is Christina saying goodbye from LabRoots, and we'll see you next time. Goodbye.